Thank you so much for you all for coming tonight. Thank you for the kind introduction, Roy. It is really a pleasure for me to be here right now. And it's so nice to see so many of you in the audience as well, considering this is the last week of the semester. So many people are very busy, including all the faculty and students here tonight. So my name is Nicole. I'm an assistant professor in mechanical engineering, as well as biomedical engineering and the robotics program and the BioFrontiers Institute, of course. And today I'll be telling you about Biohybrid Robotic Jellyfish and some of the other robots that we have in the lab. And so I'm excited to talk to you mainly about my work on Biohybrid Robotic Jellyfish, which is a live jellyfish that we attach different robotic systems to, as well as a variety of other bio-inspired designs. So I'm going to talk briefly about how we use shark skins to make more hydrodynamic vehicles, how we look at penguin flippers, how we look at manta rays, and even things with sea sponges and corals. But to motivate this work, how much do we actually know about the ocean? We know that the ocean is important for things like regulating the climate, modulating oxygen and carbon dioxide exchange, and providing us with a source of food, medicine, and transportation. But despite this, over 80% of our ocean is still unmapped, unobserved, and unexplored. And to put this into perspective, what we literally know is a drop in the ocean. So we actually know more about our neighboring planets than we do about our oceans. And it's important to be able to know more about the ocean so that we can detect and prevent situations like coral bleaching. We want to track changes in the temperature, acidity, and various concentrations of nutrients so that we can detect and prevent situations like coral bleaching, as shown with that bleached coral on the right. It's also important to monitor environmental conditions so that we can track algal blooms and make sure that we have safe drinking water for everyone and we protect all of the wildlife in these areas. And it's also really nice to just have fun and explore new species. So we'd like to discover new species such as this adorable sea bunny or nudibranch, and to discover new behaviors that happen outside of the lab. So you can think of this like if we go and take cameras into the ocean where these animals live, we might discover new behaviors that they naturally do, as opposed to when we bring them into the lab. And as an example, whenever I go home, I sit on the couch and I turn into a blobfish. That is my natural state. I don't typically act like that when I'm out in public, but I do like using this example as well because this is what a blobfish looks like when we take it out of the ocean, but blobfish typically exist deep in the ocean, so there's high pressures, and so it typically looks more like a normal fish. And so we want to learn more about animals like the blobfish and more about ocean monitoring in general. And this is a particularly good time to study the ocean because we are right in the middle of the ocean decade. So this is an international effort for scientists and people around the world to better understand and protect our oceans. And so our current methods of ocean monitoring include the following. I'm not going to go through all of these in detail, but I do want to highlight a couple of examples. So we have satellites and weather balloons, for example, that exist kind of above in that air. We have buoys and ships with various instruments, and they can be used to monitor the ocean surface, as well as do some vertical profiling. So these vertical profilers, like acoustic, wave, and current moorings, can be used to measure these vertical lines of data in the ocean. And what you notice here is that we're really good at studying the ocean surface. However, we need a lot more tools and technologies to understand what's going on in the ocean at relevant time scales, because things in the ocean are changing really quickly, as well as the spatial scales. Wow, the spatial scales, because the ocean is so vast. And so currently, our methods of ocean monitoring underwater include remotely operated vehicles, as shown here. And you can think of these like a small car that's attached via this long cable to a boat. And so this can be used to explore different areas underwater and map the seafloor. Autonomous underwater vehicles, or AUVs, can also be used, like this Slocum glider. And these offer great advantages. So they can travel fast speeds over long distances and be used to monitor the seafloor and track markers of climate change. But there's a lot that we need to learn from. So how can we actually develop more efficient and non-invasive tools to study the vast changing ocean? These traditional ROVs and AUVs can't access more sensitive areas of the ocean. So for example, if we have deep caves in the Marianas Trench, those are really difficult to navigate with these large underwater vehicles. And so that's why we look to nature for inspiration to create more efficient, smaller, and safer vehicles to send into more remote areas. So the idea is we want to create these energy-efficient, bio-inspired aquatic vehicles that can access these more remote areas, and because they swim like natural fish or jellyfish do, they would leave more natural wakes and reduce these environmental disturbances to more sensitive areas of the ocean, like near a coral reef, for example. And so we have different examples of fish robots, manta ray robots, as well as jellyfish robots here, and they all offer different advantages and disadvantages. So for example, fish can swim very fast and they're very agile at turning. Manta rays are very flexible and they're agile in terms of swimming in the open ocean or traversing along the seafloor. And jellyfish are very energy efficient animals and they don't have a swim bladder, which means that they can access areas like the Mariana Trench, thousands of meters below the ocean surface. And so we're really looking at animals as a source of inspiration for us. And so you can think of nature as a guide, not necessarily a rule book. So the earliest evidence of life, as in single-celled organisms, so tiny little cells, dates over 3.7 billion years ago. That means that these animals have had millions and billions of years to develop and have had all of these evolutionary pressures, so they're designed to operate in certain locations. So for example, this is a picture of a giraffe, and over millions of years, it has developed longer necks to reach trees in higher areas. But animals aren't the be-all, end-all. So what considerations do organisms have that robots are not constrained to? We can think of things like reproduction, feeding, maybe things like having fun for humans. That's not necessarily a priority for a robot to have. And so we can think of designing some sort of a robot, like if I were to make a giraffe robot, I would like to reach high places, but I might also want to reach low areas like this that naturally giraffes can't reach. And so this brings us to two principles that we think about when we create bio-inspired robots. The first principle is, what can we actually learn from biology? So how can we take nature and be able to figure out, okay, what do we actually need to create a robot that can do something that we'd like it to do? And the second principle, building upon this, is that animals aren't perfect. So how can we improve upon biology by using various engineering tools? And so that's where my lab comes in. My lab sits at the intersection of robotics, fluid mechanics, as well as biology. And our overall goal is to create these new tools and technologies so that we can explore more about our oceans. So to do this, we look at all three areas of research. So for example, when we think about jellyfish, we might think about the biology. So what is the anatomy and physiology of the jellyfish as shown here, but also what kind of behaviors is it exhibiting? And how can we learn from live jellyfish themselves? We also use fluid mechanics. So this is an image of a jellyfish swimming, and I'll talk more about this flow visualization technique later, but how can we actually visualize how the fluid around the jellyfish is moving? And so this graph shows the vorticity of the fluid moving around the jellyfish, and you can think of that like the swirliness. So how swirly is the water around the animal? And then finally, with robotics, how can we study animals using models? So these robo-physical models are a simplified version of a live jellyfish so that we can ask different hypotheses and be able to test them in the lab. And so to give you an idea of the variety of research in my lab, here's a spectrum of different bio-inspired constructs that we are currently studying. On the left, we have more robotic constructs. So this is an example of fish-inspired fins with little shark skin structures to interact with the flow and reduce drag and make more energy-efficient vehicles. Moving along this spectrum, we have biohybrid cyborg constructs. So this is the concept of taking a live jellyfish and incorporating some robotic/electronic controller to control how they swim. And then moving further along the right, we have more biological constructs. So how can we bioengineer corals for natural habitat restoration and other things more related to the biology side? And so I'll start by talking about jellyfish. This will be the majority of my talk, but I'll give you a little taste of other ongoing projects in the lab as well. And so before I delve into why we study jellyfish, I did want to talk about, as Roy mentioned, biohybrid robotics in general. So this is a relatively recent field. It's been ongoing for about 15 or 20 years at this point. And the idea is to make living machines. So how can we incorporate biology, that's a living organism, or take some cells from organisms and culture them in the lab, and mix that with engineering to create new tools for ocean monitoring, as well as other types of environmental monitoring. So this is a review paper. This is an image from a review paper that my students wrote last year, and they wanted to highlight all the different types of biohybrid robots that exist. So we have some flying animals, we have some walking, crawling animals, and then I'll tell you more about the swimming aquatic biohybrids in my lab. But you might be wondering, why do we care about jellyfish? Well, jellyfish are an interesting model organism because they have naturally evolved over 500 million years under various evolutionary pressures, and their body structure has actually remained largely unchanged. So they have a very simple mode of swimming. They have this contraction and relaxation, and it's tied in with all of these other behaviors. So this is how a jellyfish swims, but it's also how a jellyfish eats, and it's also how a jellyfish reproduces. So it's a relatively simple model organism for us to study in the lab. It's also really nice because these jellyfish don't have a swim bladder, as I previously mentioned, so it can access more remote areas like the Marianas Trench. And it's also really small and soft and squishy, which means that if it accidentally bumps against a coral reef, it's not going to cause harm, as opposed to more traditional underwater vehicles. And most importantly, jellyfish are actually the most energy efficient animal in the world. And we can think about this using cost of transport, or COT, as a metric of energy efficiency. So the cost of transport is defined as the energy per mass per distance traveled. What does this mean? You can think of this like the inverse of miles per gallon. So a low cost of transport actually equates to a high energy efficiency. And so this graph shows the cost of transport on this vertical axis over different masses of animals. And we have AUVs in this spectrum here. So those are those traditional underwater vehicles. We have different modes of locomotion colored here as well. And so I want to highlight that less efficient swimming is up here, more efficient swimming is down here, and what do you notice? Jellyfish have the lowest cost of transport, which means that they are the most energy efficient animal in the world. And so we might wonder, how can we create an energy efficient vehicle like a jellyfish? So to address this question, we really need to think about, how do jellyfish naturally swim? So this is an image of a jellyfish upside down. This is the top of the animal. This is the bottom of the animal. And if we zoom in on the bottom of the animal, so it's lying here on a dish, if you look around the edge of this jellyfish, there are 8 little divots, and each divot has a sensory organ called a rhopalia. And this is an organ that innervates the swim muscle, which is a ring on that underside of the animal, and it's just 1 cell layer thick. So there's not a lot going on there, but it is a very powerful swimmer. And the interesting thing to note here is that this is an all or nothing muscle activation. So even if only one of these pacemakers fires, it causes a bidirectional wave to propagate from that one location. So in order to swim straight, jellyfish can activate multiple of these pacemakers, and in order to turn, it can activate one or a couple on one side. And so in order to swim, jellyfish contract. That's called the power stroke, and that provides this active muscle contraction and a force to move it forward. And that's shown here. So this is the graph of the cumulative distance traveled over time for an animal, and the power stroke is here in white. There's also the recovery stroke. So the animal moves passively, and there's a feeding current that brings fresh water to the center of the animal where its mouth is. And that's shown here in yellow. So you can kind of think of this like a breaststroke. So you have a nice powerful stroke, you have recovery, and then you can coast a little bit too. So that's actually why jellyfish are very energy efficient. And so now we know a little bit about the biology of the animal. You might be wondering, how can we actually control jellyfish swimming? And we can think of this as an analogy by using the cardiac pacemaker. So, do you guys know what a cardiac pacemaker is? Have you guys heard of this before? So the basic idea is that the heart might need help. And so we have this electrical device that we put directly onto the heart, and that gives some electrical signal, and it creates a frequency in which the heart beats. Similarly, we can do something with these live jellyfish. So we can send an electrical signal. So here is a square pulse wave. It just shows the voltage over time. And we have certain design choices so that we can create a pacemaker for a live jellyfish. So here are the different components that go into this swim controller for the jellyfish. We have a cap, we have something for waterproofing, we have a microcontroller, which is like the brain of the robot. We have a battery, and then we have these 2 electrodes. So those are the pacemaker locations that we embed into the animal. So this is what it looks like. At the bottom, you can see a swim controller that is fully assembled, and then we embed that directly into the animal. So you see most of the electronics are in the center of the animal, connected with a wooden pin, and we have these 2 electrodes on the side. And so this is what it looks like when we embed a swim controller into a jellyfish. So here we don't have all the different components, but I wanted to show what it looks like when we have 1 electrode. So whenever we stimulate the animal, you'll see a red dot here, and the animal contracts at a more regular frequency. And then when we stop it, the animal stops moving as well. So using this system, we can actually increase jellyfish swimming up to 5 times what it would normally do. And so let's take a look at the Biohybrid Robotic Jellyfish, or the cyborg jellyfish in action. So this is our cyborg jellyfish. We have a constant flow in this tank. So naturally in the ocean, jellyfish experience ocean currents, and so they're carried passively. This is called a zooplankton. In the lab, we have to make that happen. So jellyfish get stuck in the corners of the tank. So we can't have a rectangular tank. That's why we have this curved edge here. And to simulate the natural flow in the ocean, we also have a current. So you can see some of the little particles, the food particles that the jellyfish eat, and the normal jellyfish are just being carried by this current. But in contrast, we have this biohybrid robot that's able to pulse at a more regular frequency, and it's swimming strongly against this current. So in the lab, we can run certain experiments. So this is a tank without any flow. We have a camera attached, and we have a biohybrid robotic jellyfish swimming downwards in the tank. And we can stimulate it at different frequencies. So from 0.25 hertz, which is 1 stimulation every 4 seconds, to 1 hertz, which is 1 stimulation every second. And so then we get videos like this. So this is a jellyfish with that swim controller embedded, but it's off or inactive. And this is so we can get a control measurement, which is what the jellyfish naturally would do. In contrast, we can stimulate the animal at 0.5 hertz, which is 1 signal every 2 seconds. And you can see that the jellyfish then swims 1 signal every 2 seconds. And it's quite powerful. So you have this active power stroke and a nice recovery stroke. But if we stimulate the animal at too high of a frequency, so here it's at 0.88 hertz, what happens to the animal? It looks a little weird and curled up, right? So you do get a power stroke, but the animal muscle is not able to fully relax before it has to contract again. So actually, if we stimulate the animal at too high of a frequency, then we get less efficient and less fast swimming compared to at 0.5 hertz. So from videos like this, we can then extract the data to plot something like the enhancement factor, which is defined as the stimulated swimming speed over the unstimulated or controlled swimming speed over different swim controller frequencies. And for this one animal, we get swimming speeds increased up to almost 3 times what it would normally do. For another animal, we only get a modest increase in swimming speed. And we tested 6 different animals in the lab, and we saw a clear trend that actually we can increase jellyfish swimming speeds up to 2.8 times what they would normally do, but that this really depends on the body morphology or the shape of the animal. And because I'm an engineer, I'll show you some equations, and there will be quizzes on this afterwards. So I'll go through this very slowly. But actually, all you need to know is that we have these computational or theoretical models, and the input is information about the model, or information about the animal. So how big is the jellyfish? What are the conditions of the salt water around the jellyfish? And the output is the swimming speed. So by using these equations, we can then plot things and be able to see if our experimental results match our theory. So we have certain hypotheses. And so this is the computational graph, and this is the experimental graph, and we see a same trend. So when we have flatter animals, they tend not to swim as fast. When we have these rounder, more prolate, bullet-shaped animals, they do tend to swim fast. So the shape of the animal affects their swimming speed. And so this brought me to the question of, can we actually design a more hydrodynamic shape for the jellyfish? And just keep that in mind for a couple of slides in the future. So this is talking about jellyfish swimming speeds, but I talked about how jellyfish are the most energy efficient animal in the world. And so let's think about how much power is being consumed by these jellyfish robots in comparison to other robots in the field. So this graph shows the external power per mass for different robots in the field. So how much power are these robots consuming? And this is the speed of various robots. And the marker shape tells the different types of robots. So we have more biological robots versus more mechanical robots. That's, are we using cell culture and tissue engineering for these biobots? And are we using traditional hard or silicone materials for these mechanical robots? And the color tells the different types of propulsion. So here, jellyfish swimming is shown in red. And I wanted to show a couple of different examples of these robots in the field. So the first is Jellyfish. It's a fully engineered soft robot shaped like a jellyfish. And I like using this example because the researcher's name is Jennifer Frame, and she just decided to make a pun on her name, the Jellyfish. There's also the robotic ray. So this is a biological robot made from rat heart cells in the shape of a stingray, and it's shown up here. We also have a more traditional AUV, the Remus 100, and in comparison to all of these robots, our biohybrid robotic jellyfish exists here. And remember, less power is good. So our robot uses between 10 to 1,000 times less external power per mass compared to these other swimming robots. Now, you might be wondering, okay, well, that's how much battery consumption is in this robot, but the animal also consumes things as well. So let's think about the question that is probably on your mind. Are the jellyfish hungry from swimming so fast? And so I mentioned at the beginning of this talk that jellyfish have a very simple body structure, right? So their swimming is tied into feeding as well. So every time the jellyfish swims, it creates these vortices, so these little donuts or these rings of fluid, and it brings new water into the center of the animal where the oral arms or the mouth is. And so this is nice because the faster a jellyfish swims, the faster it eats as well. So you can think of this like a marathon runner, right? He's running really fast, she's running super fast and eating that goo that is like sugar water. But anyways, the faster the jellyfish swims, the more it consumes. And it's really nice because the ocean has all of these little particles in it, and it has things like brine shrimp, which is what the jellyfish eat. And you might be familiar with brine shrimp if you grew up in the 80s and had one of those sea monkey farms. So this is basically what we grow in the lab. We have brine shrimp eggs, we hatch them, and between days 1 and 3, we feed these directly to the jellyfish. So they're fine, they're swimming quicker, and they're also eating quicker. And so this question of, well, can we bring the system from the lab into the ocean so that we can collect these markers of climate change and do other things directly in the field, brings me to the next topic, which is that we actually tested these biohybrid robotic jellyfish in the field in Woods Hole, Massachusetts. So this is a map of Massachusetts. If we zoom in here, we can see Woods Hole. And if we zoom in even further and go back in time a couple of years ago, you can see my scientific scuba diver collaborators. So we have Sean, Jack, and James. Oops, sorry. And they're in the water collecting data for me. I'm nice and dry on shore with all the jellyfish and the electronics. So I had such a rough time in Woods Hole, Massachusetts with all this field work. And then these are the buckets of jellyfish and all the different electronics because field work is really difficult. So whenever you go out into the field, things break, things might not work as expected. And so it can take a while to collect videos like this, where we have a jellyfish. So again, it's driven at 0.5 hertz. That's 1 contraction every 2 seconds. And you can see here, the swim controller is embedded and you have these 2 electrodes. So it's swimming very strongly and then it relaxes. And you can see all the fish and other things swimming around the animal too. So it seems like they are okay with this biohybrid robotic jellyfish in the ocean. And in order to track how fast the jellyfish is swimming, we have this rope in the background with different alternating marker colors so that we have a ground truth. And so by collecting videos like this in the field, we can then compare with our experimental data. So you've seen this plot before. This is the enhancement factor over swim controller frequency, and the gray data is for the laboratory jellyfish, which were those 6 animals. And in the field, we tested 2 different animals, and we saw similar increases in the swimming speed for the biohybrid robotic jellyfish. But there was a lot more variation. So if you take a look at this point, one of the animals swam really quickly and one of the animals did not. And so this is caused by natural variation, and we can go back to that theoretical model, those equations, and be able to predict how well the jellyfish will do in the field. So the ongoing work in my lab includes then taking these biohybrid robotic jellyfish, testing them in the field, and being able to incorporate things like turning maneuvers. So all of the jellyfish that I've shown you so far have swum straight. We'd like to incorporate turning mechanisms so that eventually we can have remote-controlled jellyfish and be able to send them into different areas of interest in the ocean. We also want to incorporate various sensors like temperature, acidity, salinity, etc., so that we can track what is going on in certain areas of interest in the ocean and maybe take videos of a nice little blobfish in the Marianas Trench. We also eventually want to improve the robotic design by incorporating more biodegradable materials so it's more eco-friendly and sustainable, and eventually we'd like to use even the animal's own metabolism or the natural seawater in new batteries to power these low-power electronics. And so to address some of these issues, like incorporating maneuverability and putting various sensors on, we can use jellyfish forebodies, or you can think of those like little hats for the jellyfish. And you might be wondering, what is the benefit of using a little jellyfish hat? And there are 3 that I wanted to talk to you about. So the first is that the hydrodynamic shape of the hat actually helps the jellyfish swim faster. So what I told you before about the shape of the jellyfish really affecting the swimming speeds, we can put different hydrodynamic hats on, and actually this shape is the most efficient, so it makes the fastest jellyfish, up to 5 times what it would normally do. It's also nice because we can add shifting weights into this forebody to control their direction. So can we put a freely rotating system in or a mass shifting system to make the jellyfish turn? And third, we can incorporate various sensors into the added payload. So this is a nice space where we can put things like a camera or different temperature sensors in. So we then can create more turning maneuvers with these jellyfish using a forebody. And so this animal is not turning. This experiment was just to see how it tolerated wearing this little hat. But inside of this hat is a little gimbal, which is kind of like, you know, when you see a professional cameraman and they're walking around and there's this thing on it that is protecting the camera so you get a really nice, smooth image. That's basically what we have inside here to stabilize our system. On the right, we have a mass shifting mechanism. So if you notice, there's not actually a jellyfish here. We wanted to show that the mass shifting mechanism in the hat works really well before we then incorporate live jellyfish. And there's actually another reason why these jellyfish are beneficial. Can any of you guys think of the reason? I know some of you guys are thinking it. They're very fashionable. So the non-hat wearing jellyfish in the tank are actually quite jealous because they look really good on them. So we're working on incorporating these turning mechanisms into this moon jellyfish that we're studying. That's the Aurelia aurita, but we'd also like to extend that into different species of jellyfish. So here we have a Chrysiora, which is a sea nettle, and using a single electrode, so you can see here that red light, we can actually cause the sea nettle to turn. And the Chrysiora is actually a stronger swimmer than the Aurelia aurita or moon jellyfish that we previously talked about. And you can see that here, right? There's a lot more tissue here. You have these nice tentacles and oral arms. It's eating a fish here. That's the thing that's floating away. And it's able to turn very strongly. But yeah, this is ongoing work, and the students actually don't like working with this species very much because they are nasty stingers, as opposed to the moon jellyfish. So actually, one of the things that I didn't mention about the moon jellyfish is that they do have stinging cells, but they can't penetrate through human skin. So it's really nice as a researcher to work with that model organism and not this scary guy. You do have to wear gloves with these. So our goal is to take these jellyfish, various species of jellyfish, incorporate that forebody for turning and for sensing, and eventually create a remote controlled jellyfish that can go through an obstacle course in the lab. We also then want to take this payload, put various sensors in so that we can have multiple swarms of jellyfish robots do things like map the seafloor, or in this case, map the bottom of the tank. And this is all towards the goal of putting the jellyfish in the Marianas Trench and other areas of the ocean. So how can we learn more about our changing oceans and climate change? By using swarms of biohybrid robotic jellyfish. And so this graphic of the Marianas Trench is really nice because it puts into perspective where we are, which is in that Denver area, here. And then you can see where planes fly. And then in comparison, the Marianas Trench is so deep. So it'd be really nice to have new tools and technologies to explore these regions that we've never been in before. And so if you guys are ever on campus, on main campus, actually, it would be nice to see you in the lab. So we have an exhibit that's open in the hallway, so you can see a window into the tank. I know the PhD students love it when you go like this and just stare at them doing work. They're more productive, so please do that. But we have a little plaque here that explains some of the recent work in the lab, and we have a little button. So if you press that button, you'll turn on the light for 30 seconds. So please feel free to stop by. We're always happy to chat. So one day I was looking into the tank and I noticed this interesting swim behavior. Can you see that this jellyfish is a little different from the other jellyfish in the tank? So as opposed to this nice contraction and relaxation phase and maybe some coasting in between, so this is a normal jellyfish, we had these interesting guys. So this is an atypical muscle contraction, and we like to call this hula hooping because instead of that active contraction and relaxation, you have something like this. And it just does that for hours, days, weeks. We don't really know what's going on here. And I talked to aquarium staff for years about this, and they said it's a sign of nerve damage. We don't really know what's going on, but the animals never recover from this. And I was like, really? Because I had 10 animals in that tank, and I saw that they did this behavior, and then they stopped, and they were totally normal. And I also noticed that some of these jellyfish that were hula hooping were pregnant females. So we had some nice baby jellyfish on the sides of the tank later on. So I had a few hypotheses, like maybe this is a form of swimming that the jellyfish can turn on and off, and it's a nice way for the pregnant females to release eggs or to escape from predators or other biological bases. So this is ongoing research in the lab. We don't have any published papers on this yet, so this is really hot off the presses, and I don't know where this story is going to take us, but we're interested in exploring the kinematics and hydrodynamics of this behavior. So this slide shows the kinematics. So here we have a hula hooping jellyfish, and we can calculate things like what is the frequency of this hula hooping? It's about once every second, a little faster than that. What is the speed of that muscle wave propagation? Can this tell us more about muscle physiology in general or jellyfish biology? And then we can look at things like the outer diameter and inner diameter of this animal as well. But in addition to studying the kinematics, so the motion of the jellyfish, we also want to study the motion of the water around the jellyfish. So how do we do this? Well, we can use flow visualization techniques like particle image velocimetry. And so this is a picture of our lab. I thought about taking a better photo because it's pretty, but I want to prove that we actually do work in the lab. So here we have this hexagonal tank. You can see our computer with some jellyfish in view, and then we have neutrally buoyant particles. So we seed the water with these neutrally buoyant particles that then shine when we fluoresce them with this laser sheet. So the idea is we shine a laser sheet, it's a plane, and it illuminates all of these little particles here. So you can see that it moves individually. We have a camera imaging all of this. And so traditional PIV particles are made using silver-coated glass beads or microplastics that then shine very well under that laser light. And this is really great for flow visualization techniques in general, but we are working with live animals, and these animals are filter feeders, so they'll eat anything that's a particular size in the ocean. So it's okay in the short term for the animals, but it's not great for the animals to be eating little shards of glass or plastic and then spitting them back out, right? And it's not great for the researchers either. So we have to wear these N95s that we're all more familiar with after the pandemic, but we don't want to breathe in these tiny little glass beads. And so I had this thought, there must be something better, more sustainable that we can use. And this thought came to me when I was washing my face and using jojoba beads. Hopefully some of you guys are familiar with that, but they're these tiny little plant-based beads, and they're good for scrubbing off dead skin. And so I was thinking, okay, maybe there's some sort of cosmetic or food-grade additive that we can add to the water that works just as well as these particles. And actually, we can use cornstarch. So instead of going to some scientific store and purchasing $300 worth of these glass beads, you can go to the grocery store and for $2, buy some cornstarch, be fancy, buy some arrowroot starch, those are smaller particles, and then you can put them into the water, and as they hydrate, they will match whatever density of water you put them in. So this works for fresh water and this works for salt water as well. And so then we can get videos like this. So we have a jellyfish swimming in all of these particles, and it's really pretty, like a night sky. So I wanted to show a comparison of the commercial tracer particles with our cornstarch particles here. So we're just moving a little piece of plastic back and forth to see what it looks like. And there's always something wrong with this video, but trust me, it's supposed to look identical to this, and I was gonna make a joke about like, can you tell the difference between these two videos? But yeah, that one just stops moving. So we can use PIV particles and other flow visualization techniques to figure out what's going on with the water around the jellyfish and how the hydrodynamics change between these hula hooping jellyfish and the normal jellyfish as well. But I mentioned before, our lab works in fluid dynamics. We also work in robotics. We can have these hula hooping jellyfish swimming upwards in a tank, and we can track these live animals. But if we want to simplify the system, as engineers, we can think about creating these robo-physical models. So this is a toy jellyfish. It's on a fishing line, and we have these different motors so that we can control this jellyfish, this toy jellyfish. So you can see here we have the toy jellyfish going up, and this is just a test in air so that you can see the nice comparison. It's really hard to tell with this live jellyfish, but it is slowly rotating, which is why we have these fins, and you can see the rotation better in air. And this is really nice to work with these robo-physical toy models, not just because it simplifies the experiments that we do and it makes it more controllable in lab, but also because my lab thinks about the ethics of the animal experiments that we do. And so in terms of animal welfare ethics, we think about the precautionary and minimization principles. So how can we make sure that our animals are safe, we're not doing any harm, and we're thinking about this in a sustainable way. And so in my lab, we think about the 3 R's. So the first is reduction, minimizing the number of animals, or minimizing the time with each individual animal. We also think about replacement, so the use of animal alternatives, like that robo-physical toy jellyfish, and refinement. So how can we have procedural changes in our experiments to minimize potential stress for the animals? And, excuse me, and to monitor stress, speaking of mucus, mucus secretions are a sign of stress in the animal. And we don't actually see excess mucus secretions during our experiments. So this is a sign that our animals are not stressed out. And we really think about the ethics of our experiments in terms of the individual animals, as well as the animals as an entire species. And whenever we do this field work, how can we think about the environmental ethics and the ecology? So my lab has published a few papers on this, and if you're interested, we can talk more afterwards. But we think about things like, how does our research, how do people in the general public react to our research, and how can we talk about are ethical considerations. And so in the paper that I wrote, we talk about comparable research. So this is a sea slug robot using sea slug tissues that can crawl. And then here's a robo-roach. So the idea is putting an electrical system on a live cockroach. And this is useful for neurobiology experiments. It's used in a lot of high schools and colleges to teach students about neurobiology and about electrophysiology, specifically for insects. And then there's this idea of jellyfish healing and regeneration too. So previous scientists cut these jellyfish and they actually regrew and resymmetrized. So there's a lot that we can learn about different types of insects, but we do want to think about this idea of ethics and interacting with the general public. So we don't want to play God. We are working on specific hypotheses that could have potentials for tissue engineering, human health, and ocean monitoring. And so we want to make that very clear. So I also wanted to give you a taste of other types of research in the lab. So this is going to be my series of vignettes. Speaking about more robotic constructs, we have these bioinspired robotic fins with shark skins or denticles. And the basic idea is that sharks have these microscale dermal denticles, as shown here. So this is the mako shark, which is the fastest shark in the world. And if we zoom in on its skin, you can see these scale-like structures, and they interact directly with the flow. So this is what a bioinspired denticle looks like. People previously took images of real shark skin, and so we can pattern these on these 3D printed surfaces. We can also simplify them into other types of designs, and the idea is that it helps increase the thrust of the robot or the lift as well, and helps reduce the overall drag on an underwater vehicle. So our thought was, well, can we combine the best of both worlds? Can we create a more energy efficient, drag reducing robot that uses both shark skin structures as well as robotic flapping fins? So fish fin robots have been used to decrease power costs and create more natural wakes for ocean monitoring as well. So we have a couple of different examples here, like the ghost swimmer, as well as a fish called Wanda. And so the work in our lab focuses on tandem pectoral fins, which are these flapping fins on the side, as shown here. And so we have a lot of really cool tools in the lab to study this. So this is our 2-meter tank. We colloquially call this the shark tank, because we think it sounds cool, right? And we're testing these shark skins. And you can see the 2 tandem flapping fins here. So we can zoom in, see both of the fins, see the servos. So that's how we move the fins, how we flap them, and how we rotate them. We can measure the forces and the torques on the fins. And then we can test different types of denticle features to see how much power are we actually reducing by using these textured fins. So these are the experiments that we're doing in the lab to test the tandem fins, but I also have a senior design team who just did Expo last week and did very well, but they created this really nice underwater vehicle for us. So this is a modular bio-inspired robot that can then shift between different types of fins. So we have the tandem pectoral fins as shown here. We have different space in the robot body so that we can put more sensors on, and we can also change the tandem fins to put something like a ray fin. And this is really interesting because we have a line of work that looks at manta rays and stingrays and sees how similar and how different are they in terms of swimming behaviors. So we know that manta rays are really good for gliding and flapping in the open ocean. They're very energy efficient. And stingrays are nice for swimming along the seafloor. So you see the different types of locomotion. And we had an idea, can we create a robot that has both modes of locomotion? So ongoing work, again, I don't really have a ton of results for that, but I just wanted to give a taste of the ongoing work in my lab. And then in terms of the more biological constructs, so this is the idea of habitat restoration, and can we do things with more natural organisms combined with biohybrid systems. So 2 very brief examples I wanted to highlight. One is that, how can we design artificial coral reefs for habitat restoration? So our idea is to use engineering tools to create other more natural types of reefs that we can then put into the ocean and have a nice environment for coral larvae to settle in. And so again, this is very recent work, so I don't have a ton of results, but I think that there's a lot of potential here to create these biohybrid robots, not just for monitoring the ocean, but for habitat restoration purposes. And we also have an ongoing project to use sea sponges and grow them in the lab using cell culture and tissue engineering techniques. And the idea is that sea sponges naturally produce these anti-cancer compounds, but they produce them in very small quantities, and it's really hard to keep sea sponges alive in the lab in those quantities needed to be used in human health and medicine. So we're using all of these engineering tools to create these biohybrid robots and types of hydrogels that the sea sponges like, so that we can eventually have SpongeBob save the world. So that was just a couple of different projects in the lab. We work with a lot of different types of model organisms. So again, we have stingrays, we have sharks, corals, jellyfish, penguin flippers. And then I also wanted to highlight that biohybrid robotics is a growing field. And so I'm excited to talk about this course that I developed last year, which is dedicated entirely to biohybrid robotics. So this is the first class in recorded history. And I went to an international conference and talked about it, and no one said it wasn't the first class. So I think I'm good. But it is both a lecture and a lab-based class, and it's a nice way for students to get hands-on learning with these biohybrid tools. So we have guided labs for the students, and we also have exploratory labs, where students think of their own hypotheses and work with mushrooms, mycelium. They also work with jellyfish. It's this little guy here. And then we have hawk moths. So it's a little hard to see, but we have a hawk moth antenna that we put onto a crazy fly quadcopter, and we make that drone follow different odors. So I think it's a really exciting class, and it's a great way to introduce students to the growing field of biohybrid robots. But a lot of what I do as a professor is not just about research and teaching, it's outreach as well. So I'm also happy to talk more about the Stories of Women in Fluids initiative, which is a group that was created a couple of years ago in my professional society. And the idea is that we wanted to recruit and retain more women in STEM. So we had an idea of targeting two different groups. The first is middle grade girls, ages 8 to 12, and that's traditionally when girls start losing interest in STEM in comparison to boys. And so we have this book called For All the Curious Girls, and it has all of these different chapters about the fun science that women who study fluid dynamics do, but also a good story of someone's childhood. So it's relating the science with some personal narrative. And then there's also the career anthology. It's called Persevere, Survive, and Thrive. And these are stories written by women in STEM about our experiences. And truly, it's under these three themes: persevering, surviving, and thriving. And actually my talk, or my chapter, is focused on how I hate giving talks. And I used to be really horrible at them, but I started teaching dance classes, and I found a lot of analogies between teaching dance. I know some of you guys really enjoy dance as well. And I think it's a great way to practice these scientific communication skills, and so it has hopefully made me a little bit of a better speaker. So with that, I would like to wrap it up by thanking all of the students in my lab, thanking all of my collaborators, the Denver Aquarium, all of the funding sources, and then I'd like to open up the floor for questions.